How does epigenetics turn genes on or off? Imagine your DNA as a vast library filled with books that contain instructions for your body. Epigenetics is like the librarian deciding which books to read and which to leave closed. It controls gene activity without changing the actual DNA sequence. This process is essential because it determines how cells develop and function. At the heart of this control are chemical modifications that influence how the genetic code is read. One key mechanism is DNA methylation, where tiny chemical groups called methyl groups are added to specific parts of the DNA, mainly near gene start points called promoters. When these methyl groups attach, they act like locks, preventing the cell's machinery from accessing the gene. This effectively turns the gene off. Think of it as putting a cover on a book so no one can read it. Another important process involves histones, which are proteins around which DNA is wrapped. Chemical changes to these histones, such as adding or removing acetyl groups, can loosen or tighten the DNA's grip. When the DNA is loosely wrapped, it becomes easier for the cell to read the gene, turning it on. When tightly wrapped, the gene is hidden away, and the gene stays off. It's like the librarian rearranging the shelves to make certain books more accessible or harder to reach. Non-coding RNAs are also part of this system. These RNA molecules do not make proteins but can influence gene activity by guiding other molecules to specific DNA regions or changing how tightly DNA is packed. They act like messengers or switches, helping turn genes on or off as needed. This regulation is not fixed. It can change based on environmental factors like what you eat, stress levels, or exposure to toxins. These influences can modify epigenetic marks, affecting gene activity. Sometimes, these changes can be passed from parents to children, impacting inherited traits and health risks beyond just the DNA sequence. Disruptions in normal epigenetic patterns are linked to diseases like cancer, diabetes, and other health issues. Scientists are studying these markers to better understand, diagnose, and even treat such conditions. In simple terms, epigenetics acts as a set of switches and dials that control which genes are turned on or off. It does this through chemical modifications to DNA and histones, as well as RNA molecules. This system allows our cells to develop into different types, respond to the environment, and even influence hereditary health. Understanding this layer of regulation helps us see how our genes are not just fixed instructions but adaptable elements that shape our biology.